Hello friends, this is Professor Tushar Ugale, working as Assistant Professor, Department of Agriculture.Entomology, K.K.Wagh College of Agriculture, Nashik. Today, I am going to deliver a session on the mass production of Chrysoperla carnea, that is the potential predator from Neuropteran family. Introduction of this Chrysoperla carnea. Chrysoperla carnea is known as a common green lacewing, is an insect from the Chrysopeidae family. Although the adults feed on nectar, pollen, aphid, honeydew, the larvae are active predators and feed on the aphids. Other small insects also have been devoured by this predator. It has been used in biological control of insect pests on crops. It also observes naturally all over the world. Chrysoperla carnea was originally considered to be a single species with a holotropic distribution, but it has now been shown to a complex of many cryptic sibling subspecies. These are indistinguishable characters from each other morphologically, but they can be recognized by variation in the vibration songs the insect used to communicate each other, to attract the other sex for the mating, or somehow to search of the food, especially during their courtship. Important characteristics of Chrysoperla carnea. The larvae are about 1 millimeter long when they first hatch. They are brown and resemble with the small ejaculators crawling actively around the search for the prey. They have a pair of pincer-like mandibles, or it is also called the sickle-shaped mouthparts, on their head, with which they are going to grasp their prey, sometimes lifting the victims off the leaf surface to prevent its escape. Also, a very important characteristic of this larvae to inject the enzymes into the bodies of their victims or the prey, which digest the internal organs, after which they suck out the liquidated body fluids. The larvae grow to about near about 8 millimeters long before they spin circular cocoons and pupation stage is observed. This is one important characteristic in the Chrysoperla. It is the cannibalism. Cannibalism means it is the act of consuming another individual of the same species as a food. The bigger size individuals of the Chrysoperla carnea is feeding on the less size, or the small individuals of the same species. The characteristic of adults. Adult green lacewing are pale green color with long thread-like antenna and having the glossy golden compound eyes. They have a delicate appearance and are from 12 to 20 millimeters long with large membranous pale green wings, which are the very important characteristic of this wings. It is also called as the wings with the nerve cells, that is the Neuroptera, which they fold tent-wise above the abdomen and the rest. They are weak flyers and have a fluttery form of flight. They are often seen during the evenings at night when they are attracted by light. This is one important characteristic of the green sensitivity of superpositional eyes allow the green lacewing to recognize fresh green leaves that they are used to find honeydew, which is produced by the aphids or the sucking pest, as a site for egg laying and the resting place. Taxonomy, taxonomy of Chrysoperla carnea, that is the green lacewing, is class Insecta, order Neuroptera, family Chrysopeidae, genus Chrysoperla, and species is carnea, that is Chrysoperla carnea. Morphology and biology of Chrysoperla carnea. This is the egg stage. This is the initial stage of the life cycle of the Chrysoperla carnea. Eggs are laid with a longer slender stalks. It is also known as the pedicellated eggs. Egg shape is oval. They are pale green when they are given first at the fresh eggs. Become later it turns to the grey color. The length of the eggs in various species ranges from 0.7 to 2.3 millimeter, and that of the stalk length varies between from 2 millimeter to 26 millimeter in length. The eggs are laid singly or in clusters. Eggs turn pale whitish, and then before hatching it become black. Egg period is last for 3 to 4 days. After the egg, the larval stage will emerge out from the pedicellated eggs. This is the just hatched larvae of the Chrysoperla is getting emerged out from the eggs. After emergence, it starts to feeding over the eggshell and the stalk. It is the particular characteristic of this Chrysoperla carnea after the emergence. This is the larval development of the Chrysoperla carnea. It having the three different stages of the larvae. This is the we can see how this Chrysoperla carnea larvae get developed into the fully developed larval stage. This is the characteristic feature of this Chrysoperla carnea, that when the adult the adult aphids or the nymph of the aphids is being consumed by the little Chrysoperla larvae also, it is very important characteristic that it starts feeding at a low, at the initial stage of the development. And maturity, it also consumes the different eggs of the lepidopteran pest. Even the larvae is white in color at maturity. Before that it is a brown color larvae. The larvae have 3 instar which are completed within 8 to 10 days. The larvae spines a cocoon from which the adult emerge out in 5 to 7 days. Even the golden silver color cocoons is formed by the Chrysoperla carnea. This is the pupa which is near to emergence in the form of adult. Empty cocoons are adults also observed after the emergence of the adults from that. This is the process of emergence of the Chrysoperla adults from the puparium. This is the just emerge out adult which is going to harden its appendages. This is the developmental stages as an adult from the cocoons. This is the how we are going to know the hardening process is going on after the emergence from the pupa. It get hardens the antenna, appendages, legs, even wings will get hardened in the normal air condition. This is the developmental stages pictorial presentation of the how this Chrysoperla carnea is get rid from the pupal case and its appendages is getting hardened. The step-by-step hardening of the each appendages in the adult is because its antenna is so longer, then it is not possible to just harden within a very short period of time. It will take some time. This is the again hardening of the wings, which is a lateral turn or the fully matured developed adults with all appendages. We can see the how these wings are beautiful and having the transparent nerve cells on the. This is the life cycle of the Chrysoperla carnea, completed holometabolism, complete type of metamorphosis, eggs, larvae, pupa and adult. This is the life cycle of the common green lysine, that is the Chrysoperla carnea, larval, 3 larval instar, pupa, adult and again the egg stage. The characteristics of this life cycle of Chrysoperla carnea include the holometabolous, that is the imago deposit eggs either in a group or solitary, and with the 3 larval stages we can observe the larva. Even pupation having 2 layered cocoons, it is a particular characteristic of this life cycle. Again the multivoltile, that is the 2 to 3 generations per year, is also another specific characteristic of this Chrysoperla carnea. Facultative diapause, that is the overwintering as an adult stage. Color changes in autumn from a green to brown in spring, reverse. It is the facultative diapause of the characteristic developed by this adult of this Chrysoperla carnea. Mating behavior, vibrations of the abdomen is observed to attract the opposite sex for the mating in the case of both male and female. The adults having the characteristic, like after the emergence of the adults from the pupal case, it directly starts to search its opposite sex for the mating. Generally, pre-ovulation period lasts for 3 to 7 days. Adult female starts laying eggs from the 5th day of emergence onwards, and peak egg laying period was is observed between 9 to 23 days after the emergence. The male longevity is about 30 to 35 days and female can even live up to the 60 days. Fecundity is about 600 to 800 eggs per female. The sex ratio of male to female is about 1 to 1 as to 0.85 female. The adult male and female live near approximately averagely 41 and 53 days respectively. Production procedure in laboratory. In mass production of the Chrysoperla carnea, the adults are fed on various types of diets. We have to collect the adult from the field and then we can rear in laboratory with some laboratory required for infrastructure. The larvae are either reared in a plastic tubs or empty injection vials or in a groups in a large containers, or sometimes we have to provide the individual cells or individual vials and or the rearing trays for the rearing of the larvae. The adults are collected daily and transferred to the pneumatic glass trough or GI round trough about 30 centimeter to by 12 centimeter in size. Also, before allowing the adults, the rearing troughs are wrapped inside with the brown sheet paper, that is the origami paper, which act as an egg receiving card inside the cage. About 250 adults, that is 68 may have 60% females, are allowed into each trough and covered with white nylon geogrid cloth is secured by the rubber band. On the cloth outside, 3 bits of the foam sponge near about 2 square inch is dripped in the water is kept for the feeding to the adults. Beside an artificial protein rich diet is provided in a semi-solid paste form in 3 spots on the cloth outside. The diets consist of 1 part of yeast, fructose, honey, proteinase R mixture and the water in the ratio of 1 as to 1 as to 1 1 as to 1. The adults are collected daily and allowed into fresh rearing troughs with the fresh food as prepared given above. From the old troughs, the brown paper sheets along with the Chrysoperla eggs are removed. The adults feed the food and lay eggs on a brown sheet paper inside the cage. This is the brown paper sheet showing the eggs received as a egg card on this brown paper inside the cage. Nutritional requirements to be considered while rearing up the Chrysoperla stages. Larvae requires the predatory, it is a predatory stage. It requires aphids, mites, lepidopteran. Due to it feeds on the aphids, that's why it is called also called the aphid line. It also having the cannibalism, as discussed earlier. It feeds on the eggs or the stalks of the eggs or the outer covering of the eggs. These are this larvae is very actively searching its prey for feeding. Specialized mouth parts like the sickle shape or pincer like appendages at mouth parts. Extra-intestinal digestion with some enzyme activity. It is a particular characteristic and nutritional requirement for the larval stage. Adult stage having the requirements of the food in the form of honeydew or the secretions is released by the sucking pest like aphids and pollen grains and some fungi and some algae also consumed by the adults in their life cycle. This is the pictorial presentation of the adult life cycle with including the requirement specifically for the feeding. The laying adults required feed on the honeydew, nectar, pollen, lives for 20 to 40 days averagely. Each female lays about 10 to 30 eggs per day. The laying larvae is a general predator of aphids, psyllids, mealybugs, moth eggs and larvae which is consumed during its life cycle. The group rearing of the grubs, as told earlier, it having the cannibalism. It should be reared separately from each other to avoid the damage. It is done with the GI round basin that is about 28 cm in diameter. We have to keep near about 250 larvae per basin covered with a khadi cloth. The eggs of Chrysoperla cephalnica, that is a rice moth, it is a factious host for the trichogramma and this Chrysoperla carnea rearing in laboratory, is given as a feeding material for the larvae in laboratory. For rearing of 500 Chrysoperla larvae, the total quantity of Chrysoperla eggs required is about 22 cc. That is, we have to feed with at the rate 5 cc per feeding for the 5 feedings in alternate days. 1 cc of Chrysoperla eggs is equal to 18 to 20,000 eggs per cc. The Chrysoperla larvae pipette into the round white colored silken cocoons within a 10th day after the feeding. The cocoons are collected with a fine hair brush and transferred into the 1 liter plastic container with a wire mesh covered and windows for the emergence of the adults. There is specially provisions for these plastic containers. From the cocoons, pale green colored adults with transparent lace-like wings will be emerged out within 9 to 10 days after the pupal stage. Individual rearing of the grubs. It includes the first step of larval rearing. 120 3-day-old Chrysoperla eggs is just near to the hatching. Is mixed with the 0.75 cc or 0.75 ml of Chrysoperla eggs. The embryo of the Chrysoperla eggs are inactivated by exposing them to the ultraviolet radiation, as we have seen in the tripocard preparation in the plastic containers. About the size having 27 to 80 by 6 centimeter in size. On hatching, the larvae starts feeding. After the 3rd day old larvae are transferred to 2.5 centimeter cubical cells of the plastic lowers at the rate 1 individuals per cell. Each lower can hold 192 larvae. Chrysoperla eggs are provided for the additional feeding in all the cells of the each lower by sprinkling through the modified salt shaker. Feeding is provided in 2 doses. First dose is feeding with 1.5 cc Chrysoperla eggs for 100 larvae. And second feeding is given with 2 cc for 100 larvae with a gap of 3 to 4 days is done. The total quantity Chrysoperla eggs required for the rearing 100 Chrysoperas is about 4.25 cc eggs. The lowers are secured with one side by the origami or brown color paper sheet as an egg receiver. After transfer of larvae covered with acrylic sheet and clamped. Origami brown paper is used to facilitate pupation and clear visibility of eggs. We have to make the strips of this brown paper to get maximum pupation inside the strips. The lowers are stacked in a rack 2 meter by 1 meter by 45 centimeter angle iron rack can be hold 100 lowers containing 19,200 larvae approximately. Cocoons are collected after 24 hours for formation when they get hardened by removing the origami paper or brown paper from one side. The cocoons are placed in an adult oviposition cage for emergence. The adults sometimes allowed to emerge in lowers and release on the glass window pans from where they can collect it easily with the suction pumps. This is the wooden Chrysoperla cage is also being used in laboratory for the rearing of adults Chrysoperla carnea. This is the place where we are going to keep the food in the foam for the adults. The brown paper is placed in a side for the egg laying, which facilitates the egg laying on brown paper. This is the Chrysoperla rearing plastic container showing the adults which are adhered to the nylon mesh at the opening mark the mouth of this cage. This is the flowchart which is we have discussed earlier. This is flowchart is given here with for the information of the students and for their exam point of view. This is the flowchart and very, very important for the examination. Provided the food content of the food is fructose, honey, protein mixture, water as 1:1:1:1 ratio. This is the methodology given earlier in earlier slides. The storage of the Chrysoperla eggs after the mating of the eggs, the brown paper sheet is get full with the eggs, freshly laid eggs. Freshly laid eggs is in a green color with a stalk or medicine. As such, the sheet can be stored at a 10 degrees Celsius in a BOD incubator, or sometimes it can store in the refrigerator. When the eggs are required for culturing or for the field release of the egg sheet are kept at room temperature for a day. Thereafter, the eggs during this period turn brown and hatch on a third day later. The first larvae either taken for culture, or maybe it may be for the recycling or for the field release. Storage is done by keeping the destocked eggs after treatment of the sodium hydrochloride with a 0.1% treatment solution for 1 minute, and it should be stored in 10 degrees Celsius in BOD incubator, which is adjusted for the constant temperature. The daily a sample of 10 destocked eggs can be taken out, and it can be kept outside, and the percent hatchability can be checked. This method is the best for the storage of the eggs. The problems which is being faced in mass multiplication, that is the mortality of the eggs or mortality of the larvae during the mass production. Also, we never get the good hatching rate due to the fluctuation in the climatic factors like temperature, humidity, and the laboratory cleanliness. Adherence of the eggs to the plants, which gives the low recovery rate. Even the characteristic feature of the stocked eggs, it is very difficult to get the eggs from the rearing cages. This is some problems which is faced in the mass multiplication of the Chrysoperla carnea. Application of Chrysoperla mostly done in the greenhouses. In some row crops where the specific plant to plant distance is maintained, then we have to monitor this release of this Chrysoperla carnea in the field by frequently observation or frequently scouting in the field. The prerequisites for the use of biological control as a release of this Chrysoperla carnea. The Chrysoperla carnea larvae is voraciously feeders that feeds voraciously on the all stages of its host like aphids. It is highly adaptable to the local environment or local climatic conditions, and one another important characteristic is it having the multi-time life cycle, that is complete 2 to 3 generations per year. Feed release of the Chrysoperla carnea is being released in the horticulture crops at 50,000 to 1 lakh eggs per hectare in a 3 to 5 times at a 10 days interval to control the aphids, whitefly, Spodoptera, Helicoroptera, pink balloon, thieves and mites, etc. insects. The larvae are taken in the plastic container containing the small quantity, that is the 1 to 2 cc cursor eggs, and loose paper strips. Loose paper strips avoid the cannibalism. The paper strips along with the larvae sticky and then drop in the field at random while walking across the field. Is a method given by the Ridgway and Vince in 1977. Application of the Chrysoperla carnea. The release of the eggs of or larvae. Eggs are separated from each other to prevent the cannibalism. Mostly applied by the hand spray application, that is with the help of some dispenser like equipments in the open land. Careful handling of the all stages of the Chrysoperla, that is egg stage or the grub stage or larval stage. Survival of the eggs during the procedure of. These are some products which are available in the market as for the commercial release. These products is from the foreign countries, and they having the such type of production in house laboratories. This is product prepared for the greenhouses and the polyhouses for the release against the pests like aphids and the Helicoroptera megera. So this is all over about the Chrysoperla carnea. Thanking you.